According to Stats SA 2021, it has been reported that the major cause of non-communicable diseases has been derived from cardiovascular disease, cancer, as well as type 2 diabetes. And in effect, these non-communicable diseases, they are responsible for 41 million deaths that are happening in the worldwide annually so. Therefore, there's a huge need in order to compare the phytochemical levels as well as the antioxidant activity of 5 Moringa oleifera cultivars from different geographical origins of the world. What motivated this study is that the increase of non-communicable diseases in African countries. There's a high demand of medicinal plants that include Moringa oleifera. And studies showed that medicinal plants have that ability to act as a source of antioxidant in order to fight the non-communicable diseases. And Moringa has been found to be one of those crops. And recently, several articles has been published whereby they described the antioxidant properties of this Moringa oleifera, whereas it can be translated to as the anti-aging herb. And furthermore, ladies and gentlemen, the Department of Science and Innovation is now in partnership with University of Limpopo, whereby they're initiating programs that deals with the Moringa oleifera. This study was aimed at comparing the phytochemicals as well as the antioxidant activity of 5 Moringa oleifera from different geographical origins, whereas the objective was to establish whether there is a comparative information of phytochemical levels as well as the antioxidant levels in 5 Moringa oleifera cultivars from different geographical origins. The hypothesis is derived from the objective. This study was established at Green Biotechnology Research Center, where the leaves of Moringa oleifera was harvested in April. And then the symbols were dried at 40 degrees Celsius for a period of 72 hours, whereby this dried symbol were then grinded into powder prior to phytochemicals as well as the antioxidant analysis. Here is the Table 1 that shows the different cultivars from different origins of the world, as well as their name and their code numbers, which are going to serve as my treatments. Here is the picture, the figure number 2, where I harvested my Moringa oleifera leaves. Here is the figure number 3, where the Moringa leaves were then grinded into powder. For phytochemicals as well as the antioxidant analysis, for titylphenols, the method that was used to analyze it was Folin culture method, whereas for total flavonoids, the method that was used was aluminum chloride. And then for the antioxidants, the method that was used was DPPH as well as the FREP assay. Here is the picture of the symbols where we did the antioxidant assay at the lab, and then they shows the color, which means that some symbols, they really show that they really have the ability to scavenge the antioxidant ability. As for the data analysis, this data was subjected to one-way analysis of variance using statistics 10.0, whereas the mean was separated using LCD at the probability level of 5%. Moving on to results and discussion. Figure number 5 that shows the total phenols. 5 different cultivars of Moringa, they had a significant effect on phytochemical levels. As you can see that the bars with different letters. With cultivar number 2, which is derived from South Africa, the domesticated one, showed to be the best in total phenols, outperforming other cultivars, whereas cultivar number 1, which is from Philippines, performed the least in total phenols. The same trend was observed in total flavonoids. And then moving on through to figure number 7, the DPPH figure, which shows that the effects of scavenging activity on 5 Moringa alifera cultivars. This had the significant different, as you can see that the bars with different letters, whereby again cultivar number 2, which is from South Africa, outperformed other cultivars, whereas cultivar number 1, which is from Philippines, performed the least in showing the scavenging activity. And then coming on to the FREP, which is finger number 8, it shows that the effects of ferric reducing power of 5 Moringa alifera cultivars. Cultivar number 2, which is from South Africa, it also showed that the reducing ability as compared to cultivar number 1, which is from Philippines. It performed as the least reducing agent of the ferrics. These differences, they have been attributed by the genetic makeups that are acquired by the cultivars, because some of the cultivars, they're engineered for seeds, whereas some, they're just engineered for leaves. Moving on through to the conclusion, it is then regarded that there was a significant difference in these 5 cultivars of Moringa in phytochemical levels as well as in antioxidant activity. Therefore, there was a variation in the cultivars from different geographical origins, whereas I reject the null hypothesis that says that there will not be a difference in different cultivars from different origin. And therefore, I urge farmers to adopt cultivar number B, which is recommendable because it showed the most performing in the total flavonoids, total flavonoids, as well as the antioxidant ability. Therefore, further studies can be conducted whereby they're checking other cultivars from different geographical origins of the world, where they can really look into the genetic makeup of those cultivars as well as the antibiotics. I would like to acknowledge the following: University of Limpopo for enrollment, Department of Science and Innovation for the funding and the provision of seeds, as well as NRF for the financial support. I'd like to thank you all.
Ms. Mukwena, you took 8 minutes, 30 seconds. Can I please get hands for questions? And, sorry. Can I please get hands for questions? You can go ahead.
Thank you very much. I didn't hear you clearly because you were speeding through your conclusions. With regards to the reasons that were given for the differences in terms of the analysis, you mentioned that it is assumed that the differences are caused by?
Oh, the genetic makeups of those cultivars.
Mine are comments, they are not questions, so relax. The first one has to do with your null hypothesis. When you said it afterwards, you said it correctly, but the way you put it on your slide is not correct to say there will be no variation. There will always be variation. It's the magnitude of the variation. Yeah. Then on your bar graphs, you would notice that to someone looking at the bar graphs, they look exactly, some which look exactly the same. You have got different letter scripts on them. And one would argue, why are you finding the differences? There's something which should be there, which is missing on your bar graphs. And I give you an opportunity to tell us what you think is missing. What do you think is missing on your bar graphs?
What do I think is missing?
Yeah.
I don't think there's nothing that is missing. I think everything was...
Okay, to save on time, for your bar graphs to be complete, you should have your error attached to those bar graphs. Otherwise, when you look at bar graphs which are exactly the same height, you say, how can they be different? But if you put the error there, then one would be able to figure it out.
for your presentation. I think in your conclusion you said that it's possible to grow one cultivar everywhere. In South Africa, for example, we have 9 provinces, isn't it? So the cultivar that you grow in Limpopo province, it can also be cultivated in Free State and other provinces. Is that really?
No, I think I demonstrated in my introduction that the areas where this moringa can be grown, like Free State was not included. It was Limpopo, Gauteng, as well as the Eastern Cape.
Yeah, no, I didn't really get that. Maybe you will have to clarify it, I think. And you also said further studies should be conducted. What are those further studies?
Okay, I was talking about further studies of different geographical origins of the world. Like they can have different cultivars from different origins of the world.
My apologies again. We normally prioritize adjudicators with questions, and they seem to all have woken up today and asked questions, so the 5 minutes then is over. Do you mind if I ask you to hold your question, please? Yeah, next time I'll try and prioritize you. Thank you, Ms. Mokwena. Can you please clap hands? Ms. Molabe, please come to the front. I'm hoping it's a miss. I normally get these titles wrong. Thank you. Can I ask that those who are presenting, try and stay away from Prof. Masugu there. Try and be more central just for the cameras as well. Right? So if you are presenting, try and stay away from moving too close to the crowd.
This is not my slides. Good morning, everyone. I am Molabe KM. I'll be presenting about the growth hormone gene sequence variation and its association with the growth traits of a dopper sheep. I'm under the supervision of Prof. Tiasi, co-supervised by Vimba Zima and Gunya. Okay. Here is the outline of what is expected in the presentation. In South Africa, the sheep industry is developing and it contributes to the food security and also to the economy. On this picture here, this one is a dopper sheep. It's a Martin breed that is composed from a crossbreeding Dawson horn sheep and black-head Persian sheep. A sheep is animal that is raised mainly for its meat, milk, and wool. This picture here is trying to demonstrate a growth. A growth is any act of an increase. As you can see from this one, there's an increase in growth. This process involves many factors, including the genes. In the studies, most gene that regulates growth, it was known to be the growth hormone gene. A single nucleotide polymorphism is a difference in the DNA sequence of individuals at a single point. As you can see from this picture here, these two individuals, they have the same nucleotides, however, they differ at a single point. However, there is a low growth rate of dopper sheep And also there is a high demand of heavier lambs by customers. So the breeding industry need to pay more attention in improving these growth traits that they can be used as as their markers in order to improve growth traits of different animals. University of Limpopo, where the body measurements were collected, and the laboratory work was conducted at the Department of Biochemistry, Microbiology, and Biotechnology labs, which is also in the University of Limpopo. The study used 50 animals, which are Dorper sheep, where 43 were the ewes, and 7 were rams. These animals were of 1 to 2 years of age, and they were raised extensively. A cross-sectional design was used in this current study, where there were no treatments given to the animal. Here are the body measurements that were measured at the University of Limpopo Experimental Farm. The measurements were measured using a measuring tape calibrated in centimeters, and at the same time, the body weight was measured using a weighing scale according to the suggestion of this author who published in 2012. Blood samples of 2 to 3 were collected from each jugular vein of each Dorper sheep with the help of a veterinarian. DNA was extracted following the isolation kit of this manufacturer here, and the polymerase chain reaction was used to amplify the gene. The primers that were used to amplify the gene were they were designed based on the Ncbi. The restriction fragment length polymorphism was conducted in order to detect the polymorphism where the PCR product were digested with Hale enzyme. Statistical package for social sciences, which is known as SPSS, was used to analyze the data with 5% for significance and 1% for highly significant. The PCR, the polymerase chain reaction, and the DNA sequence were used to achieve the first objective, and the general linear model was used to achieve the second objective. Here it is the results showing the amplification and restriction fragment length polymorphism of the growth hormone gene. Figure 1A, it shows the results after amplification. This is the size of the gene that was found. And figure 1B shows the restriction fragment length polymorphism, which resulted in 2 different patterns. Other animals show to have 1 pattern, 1 band, whereas other shows to have 2 bands, which result in 2 genotypes. The animals with 2 bands were noted to be AA, and the animal with 1 band, they were noted to be AB. This figure here represents the DNA sequence, which shows the position where the SNP noted. The SNP was noted on position 753. The table 1 represents the marker trait association. It was found that among all the traits, only Wither's height had association with the SNP AA and AB, where AA contributed more on Wither's height than AB. The results of the author who published in 2017 are in relation with the current study where a SNP was noted, and the study that was conducted in 2016 contradicts with this study because from all the genes that they have done, there were no SNP that was found. Results on the association, they disagree with the authors who published in 2017 because 3 genotypes were identified, and the AB genotype was the one that had positive effect on carcass weight. It can be concluded that on the DNA analysis, a SNP was revealed on the exon 4 of the growth hormone gene, and Wither's height was the trait that had an association with the genotypes, where genotype AA had more effect than genotype AB. It can be concluded that genotype AA of growth hormone gene can be used as the marker-assisted marker when selecting animals for Wither's height. And it was also noted that other studies demonstrated that Wither's height is a trait that is highly correlated to body weight, meaning if we are improving the Wither's height, we are also improving the body weight. It is recommended that the Dorper sheep with genotype AA may be selected for improvement of Wither's height, and it is recommended that more studies need to be done on the single nucleotide polymorphism of growth hormone gene using larger samples and more traits. I would like to acknowledge the following. Thank you. Here are my references that I used in the presentation. Thank you.
Thank you, Ms. Malabe. You took 7 minutes, 30 seconds. Left quite some time for questions. Can I please get hands for questions?
Thank you. Can you please explain how you design primers using Ncbi?
Okay. And on Ncbi, there are tools that are used to design the primers. Some it's called primer 3.0. It's where you're going to take the sequence of your gene and you put it on the tool, and it will drive the primers that you can use. Looking at the sequence, the tool, it will give you a sequence that has, knowing that the primers, they will bind to the sequence of the gene of interest.
Oh, in the context of selection, how do you define a marker?
Sorry?
In the context of selection, how do you define a marker?
How do I define a marker?
Yes.
A marker is a DNA sequence that has a known location in the chromosome. So a marker, it will help researchers in order to know the traits that they may improve. So in this one, the marker here is the genotype AA, meaning that if it was found there is a association between the marker and the trait. So researchers, they will use the marker in order to improve the trait.
I have to break the rule here. But you said it has to have a known location. Is that correct? Does the marker have to be, do I have to know the location of the marker in the genome, or I simply have to establish an association between the marker and a certain performance?
Yeah, sorry, yeah, you just have to know the link, to establish the link between the trait and the marker.
Okay.
Sorry, can you clarify your recommendation there?
Okay.
The recommendation, you said further, there is a mix-up there. I think the first recommendation. What did you say? What's the recommendation?
Okay, it is recommended that the dopper sheep having genotype AA should be used
Used by who?
by breeders.
Breeders. And at the same time, further studies should be conducted?
Further studies should be conducted using more traits and larger sample size.
Do you think that these two recommendations, they go together? Do you think that they are contradicting? The first part of the recommendation is incomplete.
Sorry?
The first part of the recommendation is incomplete. If you say that further studies should be conducted in traits or something like that.
I was saying it in a way that maybe there could be some other traits that are associated to the SNP, which I did not include those traits. If someone used more traits, it might be noted that other traits, they may have the association with the SNP.
Okay, thank you.
I hate to be the one that's doing this. I think my adjudicators are also contributing to a degree with asking questions and being lengthy. My brother, I'll have to ask that you hold it and ask it during lunch. This is the presenter who's presenting. Can we clap hands for Ms. Mala? Yeah. In terms of admin, I feel like I have to say this. In case of load shedding, if it so happens that we become that unfortunate and anyone is presenting or anyone is asking a question, I'm going to ask that you just hold it. We'll try and also time-wise, stop everything time-wise. And then once the backup generators fire up, then it will be an easy, restart from wherever we would have stopped, right? Okay. The next speaker is Molebala. I think for some odd reason, I fired up your presentation beforehand, so apologies for that as well.
Good morning, everyone. I am Moleba Lampo, and I'll be taking you through my research project, compiled together with my co-authors, Bofu and Mashela. The title of my project is The Host Status of Agricultural Research Council, Cowpea Cultivars to Melodigyne enterolobi, Melodigyne javanica, and Melodigyne incognita. Depicted is a cowpea, scientifically known as a Vigna unguiculata. Agriculturally, cowpea is known for its importance in the soil, which include nitrogen fixation and protecting the soil from erosion. Pea production, root knot nematodes are one of the most damaging soil-borne pests that causes a significant threat to both subsistence farmer and commercial farmers. The genus attack more than 5,000 plant species and can reduce the yield to as high as 50% to a complete crop failure. Nematicides were relied upon for management of the plant parasitic nematodes over the past century. However, due to their unfriendliness to the environment, they were withdrawn from the agrochemical market, and the use of resistance in plants have become more active as a nematode resistance alternative, and it is a suitable integrated pest management. Resistance in crops is nematode specific and should be identified using lengthy experiments. Certain crops, for example, watermelon and potato, do not have resistance to this Melodigyne species, and such crops rely on using crops with resistance in order to population densities prior cultivation. The Agricultural Research Council is researching and developing a wide range of leguminous crops, for example, cowpea cultivars, due to its limited information on the host status to Melodigyne species, along with mechanism of resistance in crops without resistance. Due to the leguminous nature, cowpea is ideal for use in crop rotations that is intended to manage population densities as a low input crops, also with economical benefits and nutritional benefits. All the potato producing regions of South Africa have high population densities of these three Melodigyne species, and currently there is no genotype among potato cultivars with resistance to the Melodigyne species. Cowpea cultivar, due to their status as a low input crops, they could be used in crop rotations that is intended to manage the population densities of the Melodigyne species, where potato is a main crop. The aim of the study was the identification of the Agricultural Research Council cowpea cultivars with resistance to Melodigyne species for future use in crop rotation systems, with the objective to determine whether the reproductive potential value of these three Melodigyne species on the 10 ARC cowpea cultivars were below 1 under greenhouse condition. The null hypothesis was derived from the above objective. The study was conducted at Green Biotechnology Research Center of Excellence, University of Limpopo, South Africa. 3 trials were established in a randomized complete block design with 6 replication and 10 treatments, with a tomato cultivar used as a nematode susceptible standard for each cultivar. These are the trials for the 3 Melodigyne species, whereby the first one shows Melodigyne enterolobi, followed by incognita and javanica. The 10 cowpea cultivars were sown in a 20 centimeter plastic pot, which were filled in with a steam pasteurized soil. The inoculum was prepared by extracting nematodes from a root of a nematode-susceptible tomato. Each cultivar was inoculated by 250 eggs and J2 using a 20 milliliter plastic syringe. Each cultivar was inoculated after every other day using 250 milliliter tap water. At harvest, 56 days after inoculation, the cowpea cultivars were uprooted and submerged into water in order to remove an adhering soil particle, with excess water being removed. The nematodes were extracted from roots per plant by maceration and blending. in 1% of sodium hypochlorite for 30 seconds, which was followed by the sugar flotation and centrifugation methods. The nematodes were then counted under a stereo microscope, which was then converted into the reproductive potentials. Data were subjected to analysis of variance using statistics 10.0 software, with the Techie test used to separate means at 5% level of probability. Table 1 shows the partitioning mean sum of squares into total treatment variation of the reproductive potential of the 3 melodigyne species. Looking into this table, it shows that the treatment effect on the treatment effect on the 3 melodigyne species was found to be greater or equal to 0.05, suggesting that they were all non-significant. With melodigyne interlobing having the reproductive potential, which is contributing 34%, followed by both melodigyne incognita contributing 31% into the total treatment variation. Table 2 shows the response of the reproductive potential of the 3 melodigyne species. Ladies and gentlemen, host status is determined by the reproductive potential, whereby the reproductive potential, which is greater than 1, means that the plant was found to be a host to a test nematodes, whereby the reproductive potential, which is less than 1, suggests that the plant was found to be a non-host to the test nematodes. So looking into this table, all the cowpea cultivars were found to be greater. All the cowpea cultivars were found to be greater than 1, suggesting that they were all host to melodigyne species. Melodigyne enteroloby was ranging from 4.61 to 209.11, with melodigyne enteroloby ranging from 11.10 to 806.80, with melodigyne javanica ranging from 51.13 to 401.50. Ladies and gentlemen, the screening trials showed that all the cowpea cultivars have the reproductive potential which is greater than 1 to all the 3 melodigyne species, suggesting that they were all host to melodigyne species, hence the null hypothesis was accepted. We also recommend that in addition to validation, other tests such as the relative susceptibilities should be used in order to assess the degree of nematode resistance in the 10 cowpea cultivars. In conclusion, using the information at hand, all the test cultivar could be viewed as not being suitable for use in crop rotations that is intended to manage the population density of these 3 melodigyne species. These are some of the references that were used in the presentation. And I would like to acknowledge the University of Limpopo for allowing the study to be conducted. And I would also like to acknowledge the Agricultural Research Council, the National Research Foundation, and Potato South Africa for funding this study. This marks the end of my presentation. Thank you.
Thank you, Ms. Malebala. That was 8 minutes, 30 seconds. Hands, please.
Thank you. Can you just explain to me, according to my very limited knowledge, you have instances whereby, yes, the roots can take up the nematode, but then there are plants that convert the, what you call it, I don't want to say the gender, whatever you call it in those terms. Dr. Pofu, Professor Pofu would know better, where you convert the sex of the nematode to one that is less detrimental to the plant. So with regards to the host status, did you take into account the ability of certain plants to convert the sex of the nematode to one that is not destructive to the plant?
Sorry, can you repeat your question?
Okay, so some plants have the ability to take up nematodes into their root system. However, some have certain hormones that they secrete within the roots that convert the female nematodes to become male, which is less destructive. did you consider that as well when you were doing your study on the host status?
No.
Hello, thanks very much for your insightful, presentations. Cowpea is one of the crop that we are, promoting as one of the most, underutilized, indigenous food crops in the country. So for food security and then, and for health-wise, as you indicated, that cowpea is one of the rich, protein-rich crops. And then that's my comment to say, continue with this study. What is your future? Because as you indicated there, this is what you can recommend, that crop rotation, which is quite very, very interesting, because we are encouraging, crop rotation as one of the, method that farmers must use, especially the small-scale farmer who cannot able to buy fertilizer and the likes. Yeah, thanks for that. I think it's very critical. But what is your future, what can you recommend for the future studies with regard to these nematodes that are hosting for the cowpea? Thanks.
I indicated on the last slide showing that in future I recommend that other people can use other methods such as relative susceptibility in order to test the nematode resistance of these 10 cowpea cultivars.
I do not see any other hands, so in that case, Ms. Malebala, thank you. Can you please clap hands for her? We've come to a point where, for those who are enjoying my chairing, it's ending now. For those who are not enjoying it, it's also ending. We have got a change in chairs. I think Prof. Signolo is going to come up and then take over from me. If you want to clap hands for me, you can, if you want. Thanks. Just as a parting shot, we know presenters are nervous. Nerves are natural when you're presenting. Please, again, stop trying to go and, stand next to Prof. Masugu there. Our cameraman is having a very difficult time trying to capture you. Thanks, Prof. Signolo.
Okay. Good morning once again. Still morning, yes. And thank you so much, Co-Chair Dr. Radebe, for steering the ship. We are moving right along, and our next presenter is Machi Munareng, who's going to tell us about morphometrical responses of the citrus nematode to Nemafrig BL phytonematicide. I hope I did justice to that. Over to you, Ma'am Nareng.
Thank you, Chairperson. Good day, ladies and gentlemen. I'm Munaring MF, here to present a study on how the citrus nematode responded morphometrically to the Nemafrig BL phytonematicide, co-authored with Bofu and Mashela. The citrus nematode, scientifically known as Tylencular semipenetrans, is classified as an R-strategist nematode due to its relatively small size, high reproductive rate, and a shorter life cycle. According to Raymond 2010, he defined morphometrics as the study of shape and size variation of organs and organisms. After the withdrawal of chemical nematicides in the agro markets due to their negative impacts to the environment, the Nemafrig BL and Nemariok AL phytonematicides were researched and developed at the Green Biotechnologies Research Center of Excellence as alternative strategies to manage population densities of nematodes. The two phytonematicides demonstrated high efficacies against population densities of various R strategies nematodes, not limited to Melodigyne incognita and the Tylenchula semipenetrans. In addition, the two phytonematicides showed the abilities to adjust morphometrics as observed in the Steinenema filtea, the K strategies nematode by Marcella and others in 2020. However, ladies and gentlemen, in Melodigyne incognita, there were no observed morphometric adjustments as reported by Marcella and Shokoyi in 2021. The cucurbitacin-based phytonematicide were tested against morphometrics of Melodigyne incognita and exhibited various quadratic relations. At low concentrations, Steinenema filtea demonstrated adjustment of body length by increasing body length, whereas diameter of openings decreased. However, in Melodigyne incognita, there was a gradual response at low concentrations of the phytonematicide, which became instantaneous as the concentrations increased. Ladies and gentlemen, as an R strategist nematode, Tylenchula semipenetrans therefore serves as an ideal nematode to achieve the aim of the current study. The study aimed to establish the morphometric responses of R strategist nematode to increasing concentration of cucurbitacin-containing phytonematicide. The objective was to assess the effects of increasing concentrations of Nemafric BL phytonematicide on the morphometrics of Tylenchula semipenetrans. The null hypothesis was derived from the objective. The source from the Zebediel citrus estate in Limpopo province, and the experiments were conducted under laboratory conditions at the Green Biotechnologies Research Center of Excellence in the University of Limpopo. The treatments comprised 0% to 64% as indicated on the slides of Nemafric BL phytonematicide laid out in a completely randomized design with 6 replications. Mature fruits of Cucumis africanus were harvested at 92 days post-transplanting, dried for 7 days at 52 degrees Celsius, and ground with a wheelie mill model 4 grinder. The ground material was then fermented using effective microbes for 14 days and filtered with a Wadman 40 filter paper. The treatment dilutions were made into 200 microliters of distilled water, and second stage juveniles were collected using the modified tray method by Wostenbrink in 1954. About 100 microliters of water with second stage juveniles were then transferred into each well plate of 96 well plates mixed with treatment dilutions and exposed for 72 hours. Post the exposure period, the nematodes were subjected to a 3-step fixation process and mounted on permanent slides for studying morphometrics. The data was collected using a ZIS microscope equipped with an electronic generating tool known as the AM scope. The treatments were then expressed as exponentials and log transformed prior to analysis of variance with statistics 10.0 computer software. Mean separation was done using the Teki test, and significant treatments at a probability level of 5% were further subjected to lines of best fit with Microsoft Excel 2016. Ladies and gentlemen, the length-related variables exhibited positive quadratic relations, whereas mid-body diameter exhibited negative quadratic relations. Using the turning point equation, the maximum values of the Nemafric BL phytonematicide concentration accrued at 3.41, 4.94, and 4.81 as indicated on the slide for body length, median bulb, and nerve ring to anterior end, respectively. The minimum phytonematicide concentration for mid-body diameter accrued at 4.5%. When body length increased as Nemafric BL phytonematicides increased, indicating that at the maximum point, the body length began to decrease, even though the phytonematicide concentration was still increasing. To explain this, The nematode comprises of muscles, longitudinal muscles in the cuticle. These longitudinal muscles work in such a way under normal circumstances that when the muscles on one side of the body contract, the muscles on the opposite side of the body, they relax. However, when the nemaphoric BL comes into contact with the cuticle of the nematode, what happens is that the muscles contract simultaneously on both and opposite sides of the body. This then leads to decrease the volume in the pseudoculum, whereas hydrostatic pressure increases. The hydrostatic pressure increase can be damaging to the internal organs that are immersed inside the hydrofluid in the pseudoculum cavity. Therefore, the nematode tends to elongate its body in order to increase the volume and regulate the hydrostatic pressure. Thus, the hydrostatic pressure decreases, and this avoids damage to the internal organs immersed in the hydrofluid. As a result, the length-related variables can be seen increasing when the phytonematide concentration increases. Similar results were observed by Marcella and others in 2020 in the morphometrics of stain enema filter, a case strategist nematode. The decrease in diameter of openings or mid-body diameter in this case was congruent with the increase in the length. In conclusion, contrary to the melodyne incognita, the triangular semipenetrans, although an R strategist nematode 2, indicated abilities to adjust its morphometrics. This suggested some degree of tolerance to the nemaphoric BL, and therefore the results of this study cannot be generalized for all R strategist nematodes. Ladies and gentlemen, the cucurbitacin nematicides had significant effects on the morphometrics of triangular semipenetrans as demonstrated, and also had nematicidal effects since after the maximum point was reached, there was complete paralysis to the nematode. Therefore, we reject the null hypothesis and recommend that further studies should be conducted to assess the ability of this triangular semipenetrans, although it's an R strategist nematode, to adjust its morphometric characters, unlike, instead of behaving like the R strategist nematode melodyne incognitum. These were the references used to compile the presentation. In closing my presentation, I would like to acknowledge the National Research Foundation for funding and the University of Limpopo for enrollment. Thank you.
Thank you, Ma'am Naring. You've used almost your time, 9 minutes, 30 seconds. May we have questions and comments for Maji?
Thank you, Ms. Monaring, for your wonderful presentation. So I need to ask, what's the uniqueness of your study compared to those you were comparing to, the likes of Marcella et al. 2020?
Marcella and others conducted a study on a case strategist nematode assessing the morphometrics, how morphometrics responds to nemariok AL and nemaphoric BL to the increase in concentrations. Although similar increasing concentrations were used in this study, triangular semipenetrans is an R strategist nematode, which is different from K strategist nematodes, and also it is a citrus nematode. And stain enema is an entomopathogenic nematode.
Okay, thank you.
Thank you.
Thank you for the presentation. In your methods, you mentioned that you used a method that was developed in 1954. So my question is, are there no new methods?
Well, recently there's been a methodology that was done by Shoko He. However, he hasn't published the paper for the sake of referencing. Therefore, the method is similar to that one that has been used in 1954 by Ostenbrink. Hence, the citation.
Do we have any other question from Majie? Okay, it looks like we do not have any further question, Majie, and comments. Can we give Majie a round of applause? Our next speaker is Kabelo Nuku. Nuku is going to tell us about the small-scale maize farmers' perceptions and adaptation towards climate change in Makhuditamara local municipalities, Kukuni district of Limpopo province. Can you assist the presentation? No. Thank you. Thank you, Dr. Radebe. Over to you, Ma'am Nuku.
Good morning, ladies and gentlemen. I greet you all. I go by the name of Noko Kabelo, and I'll be presenting my research titled Small-Scale Maize Farmers' Perceptions and Adaptation Towards Climate Change in Makhuditamara Local Municipality, Sekhukhune District, Limpopo Province, under the supervision of Dr. M.A. Nkwana and Prof. A. Belete. This is the outline of my presentation. The problem statement. The agricultural sector is the most important driver of economic growth in rural communities of the most developing countries. When we look at the agricultural sector, basically in South Africa, it is the one that provides more employment for rural people. And when we look at maize production in South Africa, maize is a stable crop that we use on a daily basis. And most farmers tend to farm maize in rural areas, whereby they sell it for productivity and also for stability. And climate change in this study, as citing IPCC, according to IPCC, which is Intergovernmental Panel of Climate Change, they define climate change as a change in weather patterns through high temperatures and low rainfall. So the main factors influencing perception and adaptation in this study are socioeconomic factors and institutional factors and contextual factors. When we talk about socioeconomic factors, we talk about age, gender, level of education. And when we talk about institutional factors, we talk about the government and extension services, for example. And when we talk about the contextual factors, we talk about crop failure. So for farmers to adapt, they tend to perceive first that climate is changing. So to this extent, farmers are mainly affected by these particular factors. The aim of this study was to analyze the small-scale maize farmers' perceptions and adaptation towards climate change in Makhuditamara local municipality, Sekhukhune District, Limpopo Province. And the objectives were to identify and describe small-scale maize farmers' socioeconomic characteristics in the study area and assess the level of perceptions of small-scale maize farmers towards climate change in the study area, as well as to analyze the socioeconomic factors influencing the perceptions and adaptation of small-scale maize farmers towards climate change in the study area. The research hypotheses are in line with the objectives. moving along to the methodology of the study. The study was conducted in Makudutamaga local municipality, Sekhukhune district, mainly the two villages selected, which is Appelcross and Ramasemola. And the sampling procedure that was used was qualitative and quantitative data in form of questionnaires and also literature and journals, articles, and so forth. So the primary cross-sectional data was also used, and simple random sampling was used to select the farmers in those villages. And also the SPSS version 26.0 and Excel were used to analyze the data. So in this table right here, it's it's a demonstration of how we came about the sample size. So our sample size was 110 from a sample frame of 238, whereby in Appelcross we interviewed 43, and also in Ramasemola we interviewed 67, making up the 110 small-scale maize farmers. And the methodology, the methods that were used to address our three objectives was that descriptive statistics using graphs, frequencies, and so forth were used to address the first objective. Likert scale method, whether the farmer agreed or disagreed or strongly disagreed that climate is indeed changing. Based on the two components, I forgot to mention that earlier, based on the two components, which is temperature and rainfall, we used Likert scale, and also our model, which is Heckman two-stage simple selection model, was used to analyze the data. This is the Heckman two-stage simple selection model, whereby our equation one, it is the outcome stage of the model, whereby the farmer adapts that climate is changing. And our equation 3 is the selection stage, whereby the farmer perceives that climate is indeed changing. So for the farmer to adapt that climate is changing, they need to perceive first. And then this is the continuous variable description of the small-scale maize farmers, whereby we have the significant variables, age, household size, farming experience. So in this table, I'm just gonna briefly give you an example using age and farming experience, whereby the mean for age was 58 and the minimum was 31, the maximum was 85. Farming experience was 31. The minimum was 7 years experience and the maximum was 58. So this shows that the older the farmer is, the we have in farming. So according to Mandisa and Deresa, in their study, they outlined that age goes hand in hand with farming experience. So the older the person that is in farming, the more experience they have. So a person, for example, I will give example with my co-supervisor right here, Professor Belete, looking at his age, if, for example, we can say that he's a maize farmer, he has more experience and he knows the climatic conditions that have been occurring for a couple of years that he has been farming that particular crop. And looking at myself, in my late 20s to him, so hence that's why we have the T-test, significant two-tailed level of significance that shows the relationship between the variables. And these are the categorical variables, whereby we have the marital status of the sample scale maize farmers, we have the employment status and the educational level. I cannot go deep in those, but we can see by the graphs how they are. And now we have the continuation of the descriptive variables. We have the number of cold days perceived by the small-scale farmer during the winter season in the last 30 years. We have the perception of temperature, number of hot days during summer by sample-scale maize farmers. And we also have the perception of rainfall, number of rainy days during rain season by sample-scale maize farmers.